Hello, this is Mrs. W, and this is a short video on how to solve a simple monohybrid genetics problem. The genetics problem we're going to solve today is frizzled feathers is dominant gene in chickens. A smooth feathered chicken is crossed with a frizzled chicken that is heterozygous. What is the expected genotypic ratio of the offspring? All right, so first thing I like to do when solving a genetics problem is figure out what the possible alleles are. So remember, you're usually looking at least two possible alleles because we can inherit one from our mother and one from our father. It's possible that we have two different alleles or they can be the same. So in this case, we know that frizzled is dominant, so I'm going to assign a capital F to represent frizzled. And a lowercase f to represent smooth as our two possible allele types. One of the other things I like to do really quickly is to go ahead and figure out my possible genotypes and phenotypes. So if I have the genotype capital F, this is going to represent a frizzled, and I'm just going to abbreviate here, chicken. If I have capital F lowercase f, this is going to also be frizzled because it has that dominant gene. And then if I have two lowercase f's, that is going to be the genotype homozygous recessive, which would be smooth. So just in case you missed it, capital F is homozygous dominant, frizzled. Heterozygous, capital F lowercase f, means that I have a frizzled allele and a smooth allele would be frizzled feathers, and then homozygous recessive would be smooth. Now to go ahead and figure out what my cross is going to be for this case. Well, I have a smooth feathered friend here, so we're going to have two lowercase f's, and then a heterozygous frizzled would be capital F lowercase. Next thing I need to do is set up my Punnett square. Now what happens during meiosis is these two alleles are going to split up during meiosis 1. So let's take a look at an example of that. If you're looking at this picture of meiosis, let's assume that the larger chromosome is going to be where the alleles are for feather structure. So I'm actually going to have us look at the frizzled ones so that we can see the difference. So let's assume that blue is the paternal or the chromosome that came from the father. Let's say that that's where the capital F is from, and that the red is going to represent the other one, which came from the mother, so that would be the lowercase f. Well, after meiosis 1, you can see first thing we make an exact copy, so we have an exact copy attached. After meiosis 1, you can see that they are split up into two different cells, and then you can see that split again. So that's what we're representing with the Punnett squares. We have to figure out how many possibilities can we get from these two chickens mating. And so these are going to be split up into two different cells after meiosis, and so we're going to have a capital F and a lowercase f possibility. For our smooth chicken, they have the same alleles, so it's just lowercase f, and then we do our cross. If this cell, this gamete and this gamete fuse together during sexual reproduction, I'm going to have capital F, lowercase f, and then you do the same. If these two fuse together, I'm going to have capital F, lowercase f. Keep going. Lowercase f, that's if these two fuse together, and same thing here. If these two fuse together, I'm going to have lowercase f. So you can see here that I have two heterozygotes. So two heterozygotes, and I have two homozygous recessive. So this is a 2-to-2 genotypic ratio for this particular cross. Hope this helps.